This video is brought to you by Vita Bee Health. Thanks Vita for sponsoring this channel. Please check them out, link in the description of this video. Hello everyone, this is your host Dr. Umberto Boncristiani, and in today's episode we're diving into the fascinating world of good bugs versus bad bugs with none other than Dr. Jay Evans from the USDA Honeybee Research Laboratory in Beltsville, Maryland. Dr. Evans is one of the pioneers researching bacteria in the gut of honeybees and his relationship with honeybee health. Today, his work focuses on understanding the complex relationship between bees and their pathogens, as well as the pests that threaten them, like varroa mites. In this episode, we'll explore his innovative approach to promoting bee resiliency and balancing the battle between good and bad bugs in bee ecosystems. So stick around for a deep dive into the science behind safeguarding our pollinators. And now, I introduce to you Dr. Jay Evans. Hey Jay! Welcome back to the show, my friend. How are you?
Thank you, Umberto. It's really great to see you again and be on your show. I'm honored to be here.
Yes. I think the last time that you went to the show, I know there is one time that I was in your house, that I visited you in your house and we recorded that there. But I don't remember if you came to the show after that.
I think that might be it. That was, and that was in the winter, I think, and we were discussing the challenging topic of bee losses, and it was sort of
CCD.
So CCD. Maybe we can be a little peppier today than that.
That was that was controversial.
Certainly, honeybee colony losses are always on both of our minds, and just how tough it is to be a beekeeper these days. So
yeah. We both of us are concerned about the industry, work to make the industry stronger. So when those controversial topics arises, we need to talk about them sometimes. But that's not the case today. Well, maybe. Let's see. Today I want to talk about microbes in honeybees, and you are one of the pioneers in this field, Jay. You start some work with that a long time ago. I remember one of the articles that got my attention was in Journal of Economic Entomology. I think the title was Bacterial Probiotic Induced Immune Response to the Honeybees. And that starts my understanding about this whole journey. And so it starts for so long ago. How do you see bacteria interfering with bee health?
Yeah, so it's a
very broad question. Yeah,
and I will go back a few years before that. I was hired by Dr. Hachiro Shimanuki, who was an expert in American foulbrood primarily and other bee diseases, and he was keen that I study foulbrood. So I did spend a couple years, and then a little bit after that, but kind of focused in on that disease. Mites were certainly an issue, and were being addressed by researchers in the lab. But it is still a persistent disease of honeybees. And I also thought it was a nice system to get going with bee disease, and especially how bees can fend off bacteria like the agent behind American foulbrood. So I loved working with foulbrood. We had a great setup where we could inoculate larvae in the lab, follow them over a few days, and then conveniently we also had a field apiary that Dr. Jeff Pettis had set up, where we could actually do inoculations and experiments outside in real colonies, and it was heavily quarantined, of course. So there were those tools available. It was a tractable disease. I've since, as you know, joined the bandwagon of viruses and mites and other severe challenges of adult bees. And that's mainly because I do think those have a higher economic burden for beekeepers. But bacteria are fascinating. So it started, we did some genetic experiments looking at how bees turn on an immune response to bacteria, especially the American foulbrood agent. And kind of getting a sense for what the bees could do on their own to battle this. At the time, the lab was developing the two currently licensed new antibiotics as well for use against bacterial disease. And so it's kind of a balance to that to see, okay, well, we know the beekeepers need tools, the regulators need to prescribe tools for this. But what are the bees up to? And my background was more on the bee side than microbes. So I wanted to find a way to look at bees. So we did some now pretty simplistic experiments, exposed them to a few spores of bacteria early in the first instar, and then just freeze the bees after consecutive days and see if they're turning on a response. And in fact, they do. They turn on kind of a classic innate immune response in response to bacteria. And we did one project with Laura Decanini and Anita Collins, which still it's kind of lost in the histories of time, but it was one of my favorite projects. Anita was a great quantitative geneticist, so she did some single drone inseminations of bees, and we were able to set up 40 different patrolines from the same daughter or sister stock, and really fun experiment. Everything worked pretty well the first year. The second year, we didn't get much queen. We tried to carry it on to multiple generations, and in short, found our seasons were short and time was short for researchers. So it was a one-off kind of study one summer, but it worked really fun. We got to see how very strong differences in different bee lineages in their response to American foul brood. So there's clearly a genetic response at the individual level to that bacterium, and that could be exploited for breeding. So that was really exciting just to see the bees were handling it in some cases well, in some cases not so great. One of the offshoots to that, which actually led to the paper you're talking about and some other work with bacteria, is we thought we were trying to save money, I think, more than anything. And so instead of these somewhat expensive genetic tests for immune responses, we resorted to kind of mashing the bees on a media plate, which is basically food for bacteria that had been coated with the P. larvae bacterium. And to our excitement, sometimes there was this inhibition around those mashed first and second instar larvae, and we thought, oh, those are super immune bees. But if we let them go a couple of days longer, we noticed that there was actually another microbial growth around the larvae at the same time. And this was work with the then Don Lopez, who should be a doctor for all the science she's done in the bee world, and Tamika Armstrong, who was an undergraduate. So what we did is to pick out those other bacteria in those plates and culture them. So we grew them up, and they were kind of now known as bee bacteria from Bacillus primarily, and Brevibacillus. And it was striking, though, that it wasn't the bee itself. So we could take those bacteria, grow them up, and put them back in concert with the P. larvae bacterium, and they just blew doors on the bacterium. They cleared the plates better than the bees had done, of course. So we knew it was the bacterium fighting another pathogen, basically. And that, to me, kind of piqued my interest in this world that we'll talk about soon, of sort of probiotics, or good bacteria, bad bacteria. We did do a couple field trials with those in this apiary with American foul brood. And at the time, I was sort of thinking it's got to knock it out because it's such a deadly disease, and we really need strong effects. And it didn't do that, but it did knock the foul brood levels down across. We did 13 different trials, basically frames of brood given the pathogen and or a whole array of native bacteria for these. And in 13 trials, there was a lower P. larvae level and lower mortality, actually, in the ones given this sort of native bacterium. Sometimes it was just a bit lower. It was not necessarily a real, it wasn't a knockout punch in any case, really. But it did seem to have an effect. So that was exciting. We however, decided that might not be enough for American foul brood, which you don't want 30% less of, you want 0 of if you can get it. And so gradually kind of, published some work and then moved on to other things, I guess. But
so let me interrupt you a little bit. So just for me to process, So this is one of the first demonstrations that bugs were fighting bugs with bees, basically, if I remember correct. I don't know if the first one, but one that they have a very big impact because we were talking about a very important disease of the honeybees. So we know for a fact that bugs are able to interact with bugs, and a lot of different things can happen in that field. And you found, and it was very significant finding, that another bug could basically help. And with that being said, do you know more of these cases today, or where we are in that field about how bacteria or other bugs can actually help against a pathogenic bug in the honeybees today?
Yeah, well, in honeybees, there's been a lot of similar work. I think some with chalk brood now, also with the foul broods. And it does seem, whether it's competition or direct warfare between those bacteria, there is an inhibition, and not all, of course. I mean, we did tens of bacteria and really found a few that looked like they were consistently inhibiting growth. So it's very true. And you think of it in any system, whether it's the soil or an organism, most bacteria are kind of their enemy, in a sense, is each other. So there's things they produce, they secrete, they cover the substrate, whether it's a gut or a plant cell, to keep out their competitors in some ways. So this is not new, and it was new to me. I will say I wasn't a real microbes fan at the time, so I was learning a lot. I was like, wow, this is where penicillin comes from, and all these stories we knew of, but they really are bugs fighting other bugs most of the time.
And they can do other things too, not all fighting bugs, but we know there is some evidence they help with digestion. There is a lot of different things that can happen, right? In that microenvironment.
Exactly. And that's what you're leading to, and what you've shown and talked about, and I think is beautiful, again, a whole field unto itself, is the activity of the microbes in our own bodies, activity in the microbes in bee bodies or on plant leaves, often is beneficial for the host, or at least benign. Our lab is 95% focused on the bad actors, the nocemas, the viruses, the pathogenic bacteria. But ounce for ounce, or cell by cell, most of the microbes in bees' bodies are either hitchhiking, in a nice warm environment, or in many cases, they've evolved to benefit their hosts. They're often they're obligate to that host of the bee gut, and over millions of years, that can lead to those bacteria being actually, kind of figuring out ways to maybe being co-opted by the bees. But I think most of the time, bacteria that succeed are in it for themselves still, but they're figuring out mechanisms where their growth doesn't negatively affect the bee, but it actually can help it. And you're right, there's nutritional examples that abound that suggest that these bacteria are actually taking on some of the digestive roles, maybe, of bees.
My point is that it's so many variables on those things. And as a researcher myself, I want to ask you how we narrow down to find the things that can be useful, in all those potential interactions that happens every day. How can we narrow down in a research base to find the interactions that might be good for the beekeeping industry, and in which time, because we need to consider time too. Some things that happen in 1 month might not be able to happen in another month, and vice versa. I wonder how you tackle this research question, how I choose my projects to narrow down as efficient as possible.
Yeah, that's a good question. And most of our efforts in Beltsville there are directed towards mites in the case of Dr. Stephen Cook and others, and viruses lately, and nosema. And we pare it down as far as we can get. We're trying to keep it usually a a whole bee involved instead of the cells of a bee currently, but that's another fascinating avenue. But we will do very controlled infections of those bees, somewhat like those foul brood infections, where we knew we were giving them, a lethal dose after 3 or 4 days. So we'll do controlled infections and then just test one little thing at a time with a bunch of replicates of that one thing, and that's a way to narrow down a pool maybe of bacteria in this case that are good for bees. As you hinted, it doesn't apply to all seasons or maybe any season of being outside in a beehive. And that's a big challenge. We've had medicines that we were super excited about that haven't translated to colony health. And, we're missing something there. And that's the complexity of the hive, the different activities by the bees themselves, food they're bringing in, maybe stuff like that. But yeah, so the first step is usually much reduced experiments, one agent, one treatment at a time. But what you really want is to get a soup in some sense.
Jay, we know in other fields, like the human fields, that, that are, for example, probiotics are very easy to demonstrate today. There is some benefit. And going to the bee field, we still I believe, that's my position right now, in a gray area. There is some indication that can be good. There is some indication that doesn't do anything. I didn't see any indication that is bad, but we are in a very gray area comparing with the human research, for example. I wonder your thoughts on that. Where do you think we are in that gray area? Do we have examples of good and bad? And after that, I want to give my personal experience with some field trials that I did myself at University of Florida, and I want to discuss this with you.
Sure, yeah. And boy, the last 2 decades now, almost, maybe 15 years, there's been a huge resurgence in microbes in bees other than pathogens, really triggered by a lot of work by Professor Nancy Moran now at the University of Texas. And so we went from, again, when we did those foul brood studies, we would sort of grow things up and jab them and stab them and freeze them and try to see what they were. But we now have really complete sort of communities of the bacteria in the bee gut that are pretty well known, characterized at the gene level and everything. So that work has made a great library toolkit, as it were, to build from. So we have all that. Her group, other groups are of course interested in how that affects bee health, and maybe giving it a boost somehow. And that's where probiotics and prebiotics come in. It's like, if you understand that system and you understand your pathogen, how do you marry the two and try to improve bee health? And our lab has helped with some of those trials. We are not really driving any bus like that. There's been just fantastic work at the industry level and at academics like Dr. Moran and USDA labs in Tucson, Arizona. There's been some really nice work. So I think that's the future, honestly, is to try to find some of these resident bacteria and to work with them and work with the bees to help with bee health. But it's an exciting field. It's a very dynamic field right now.
Now it just come to my mind, and before I give my experience with the probiotics, just something that crossed my mind from our conversation that we have a long time ago. Back in the day, I think like 15 years, 17 years ago, Jay, I remember we having a discussion that you mentioned that you were seeing. We know that we have the new vaccine for honeybees today against Bacillus larvae. And back in the day, I think you mentioned that there is one of your experiments that you did have suggested something similar happening, but you never pursued. But do you remember any of these things going on in your mind?
Yeah, the closest we came to something like that was within a generation. We didn't successfully do
the transgeneration
that went from queen to offspring, and that's a fascinating field. There's been scatterings of successes in different insects doing that. So ours was more sort of triggering with, and we were using surrogate bacteria. We were using a lactobacillus blend, basically something we bought at a health food store.
Yeah.
And showing that you could prime the immune response in that way, and those bees would have lower disease loads later on within that one bee's life.
One life cycle. Yeah. Very, very interesting.
Yeah, and that was really fun. And so I am a believer that bacteria can somehow tickle the immune response to the hopefully benefit of the bees if they're then exposed to disease. The challenge is, of course, none of us want our immune systems raging all the time. So it's something that's best when it's followed, when it's predictive that you will have a disease challenge coming up, I guess. But yeah, it's a great, fascinating field. And we,
So your thoughts on the vaccine that we have today? There is a long way to go, I believe, but I also am a believer. I believe there is it might work. It might work pretty good. The vaccine itself, the company, they have some trials already, get a provisional license, and yeah, I am a believer. So I'm looking forward to see more data on that, especially directly from beekeepers, what they say. So I'm really into this. Looking forward to see more of that, to listen more directly from the beekeepers now.
That's where it is right now, and that's going to be fascinating. Just in the field, relatively controlled experiments,
real life experience with the beekeepers.
Great, any advance to bee health is a good one, and hopefully, this will be such an advance. I think it will. Needs a bit more ground truthing, I guess, in the hive, but it's the scientists who work this out are very good. I think it's more, again, that sort of challenge that we all face of something we get super excited about in a controlled setting, and just you kind of go on a wing and a prayer sometimes to hope it will work in California.
Yes, that's it. Good. Let's go back to probiotics, Jay. I want to share. I think it's the first time they're sharing this in a recording setting, but I had a couple of trials that I did. Actually, it was 2 trials. One in University of Florida that I did myself, and another one that I did it in my consultant business for a beekeeper. So. And these 2 trials that I did, I used probiotics available in the market. And in one trial, I didn't get any result. But the one that I did at the University of Florida, I got an amazing result. However, as I said, I believe we are in a gray area with some of those products that are. And the product that I used was what's the name of the company? Strong microbials. The one that I use was strong microbials. And at the University of Florida, I got the bees was much heavier than comparing with the controls. However, there is a problem. It was in the middle of COVID. That's why I don't say too much about that all the time to everybody, because it's not a done deal to me. I put the probiotics, and I know the probiotics have some sugar in it. And it was in the middle of a dirt, and was in the middle of the COVID, and I lost all my students. I need to do everything myself, so I need to reduce the amount of data that I was collecting. So I got a little lost in the details there because of COVID, but I cannot deny that I got the result. It was statistically significant, more than 30 hives per group. It was a good trial, but I don't know if it's the little sugar that was already in the product or the bacteria themselves. So I'm kind of, man, I didn't control this very well. This could be a good result that we can show to the beekeepers. But I need to tell that in another field directly with the commercial beekeeper, I didn't get anything. I wonder your thoughts on the probiotics like that. Have you what do you see in the field? Did you hear anything about that? I wonder about your thoughts about my experience.
Sure. Yeah. And the usual disclaimer as a federal scientist, I don't approve or disapprove any commercial products.
Oh,
I
let me
think
go back to this statement. We gotta go back.
people are trying hard to help bee health. I'll say that.
Yes.
And at the commercial level, obviously the beekeeper level too, and the researcher level. So I think your result, from those two efforts probably reflects that, environmental conditions, needs of those bees. Are they nutritionally stressed? Are they facing a disease like nosema? Are they facing a pesticide? And that's where it comes down to, is until we get a really fine sense for any product, what its mode of action is and when it might be applied. I think there will be many cases where there won't be an effect. To the credit, and I've heard this from all of the, I would put the probiotics with other supplements, other than the miticides, which obviously clearly have a very strong positive effect, but can have adverse effects at treatment time. All of these sort of, the ones that I know, but the sort of feed supplements are not harming colonies. They're not, toxic to bees or to ourselves, and that's a good thing. There are experiments, and there just need to be more experiments, but there are some suggesting that in routinely applied trials, there's not a huge change in bee health with them. So it's back to not, you're not hurting your bees. It's back to an economic argument. If the beekeeper is able and willing to invest in a product that could work, but maybe often needs a bit more vetting. That's halfway there. I mean, to me, it means that, these are you're putting live microbes into a colony with a predicted mode of action too. I mean, they're not willy-nilly, and not harming the bees, certainly. And maybe there is something that wasn't measured at any given trial that was a threat, like a virus or a gut parasite that was reduced. We can't measure everything as researchers, and historically people would simply measured honey yields at the end of the year and things like that. Yeah. And that's a good surrogate for a lot of the hard work of measuring pathogens. But that doesn't get at, was there a risk of foul brood or going into the winter or chalk brood the next spring, which is, I think a lot of the probiotics, if they're working, are going to be subtle. They're going to have a kind of a film almost in the bee gut that keeps the risk of, even your beloved nosema into the gut. So that I think the risks will not be declared, or I mean the benefits rather will not declare themselves because the risks aren't universal. But I'm certainly not opposed to that line of both research and industry. I think there's a lot still to learn about it.
The way I see it now, Jay, where we are now in my position is that I don't think we know. Actually, I don't think we know more about mites than anything else. But in every tool we develop in beekeeping in the honeybee field, we need to nail down the quantity, the strain, and the time. And those things vary dramatically sometimes, and sometimes you don't get the same things that we are able to do in a lab repeatedly in the lab. Okay, we get this done, but then we translate that to the field, things never happen. And I think it's based on this triangle of circumstances, quantity and time, and the strain of the bees, or the strain of the microorganisms that we're working with. I'm optimistic about this field, and I think it's a matter of time until we nail down, okay, this is going to work, but you need to use it this way. Follow what I'm saying, this specific way at this time of the year with this bee, blah, 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 blah. So that being said, because I was bombarded with emails and things like that, I need to ask you and put you on a highlight here, Jay, and you answer this question if you can. There is a harsh letter from USDA, from Kirk Anderson, about probiotics in ABJ, I believe, or I don't remember the B journal. And I want to ask you if that's the official opinion of the USDA, or if it's because I was surprised because because I work at USDA and I know the rules there. I wonder your thoughts about that letter, and you answer if you can.
Okay. Oh, that is a hot seat. No. So, and I've known Kirk for probably 15 years, and I've known his science.
Yeah, it's great science.
Fantastic scientist, and trying his best to work with, as were his co-authors, to work with beekeepers and get relevant data. Yeah. So that the paper from which that letter highlight, I guess, derived, was very sound science. It was done in one environment, equivocal results on some colony health metrics for a couple, I think a couple commercial probiotics. Perfectly well represented, great paper. Yeah, Kirk is a very direct person. I know that he wasn't speaking in the letter on behalf of USDA. We're not actually. He's in the same branch of USDA as myself, but we're not a regulatory group or a research group. So I don't think he was pretending to speak as a regulator.
Okay.
And so I think that might not have been clear, but I think he and I think he's moving on. He's trying to test all these questions as well. So I guess
I don't have problems when people have opinions. And by the way, I will invite them. I will invite Kurt and Randy Oliver to the show. I hope they accept the invitation so we can discuss the findings and the letter itself. But my understanding, the way I felt in the field, working, trying to improve the field, is a USDA representative. There is a lot of companies that are trying to do good. There is a lot of companies that try to do bad, and I feel this is the work of the regulatory people, and that could create problems. But I know it's my opinion. I'm gonna bring them on in a show, so I'm gonna give the space for them to talk. And I'm working on a video series about probiotic research, so we can put all this data together, and everybody else in the field can jump in and give their opinion, because I think it's important. The more we discuss about these things, easier it is for us to get the right path, where we're supposed to go with the research. Maybe we miss a couple things here and there with other research, or some controls we're missing, or some data we're believing at the time, but now maybe you should not believe that too much and move to a new direction, things like that. So that's why I put you in a hot spot, Jay, because I was bombarded with these things, and I was thinking that cannot be a USDA official position.
Yeah, and again, it would be great to speak with them and others involved. It's especially about the research. I think that's primarily, I would hope, the message you wanted to bring across is their own study that was completed.
Yeah. Well, sorry I put you in a hot spot, Jay. I mean, let's move on with this. I wanna know, what are you doing right now? Are you doing with some bacterial things right now? I remember there's some natural products going on. Is that related with bacteria too?
Yeah, it's not. It's your other twin passion as a researcher, viruses. So we have joined that effort of now many researchers to really get the first and most effective antiviral medicine for bees. And we are lots of candidates, I would say, hundreds that we've tested. Some really exciting lab moments where we see something.
Yeah. Anything that can be shared?
Well, not only because we don't have a field confirmation of these.
Okay.
We just can't seem to get that bridge. We've tried different deliveries. We're really into fondant these days.
Yeah.
All that. And I always say I'm an optimist, so I think, come back in a month and I'll tell you how this fall season goes. But I know that we have compounds that are inhibitory for, in this case, newborn wing virus, and that was our goal. And our primary focus has been on natural products, which is a pretty broad group. It can be from mushroom mycelia to nectar compounds to pollen compounds to soil bacteria products from those bacteria. Our hook, and it's really because of colleagues, is on the plant side. So we're really keen on things that bees pick up in nature, in some cases at will, because that plant happens to be flowering. In other cases, they're unlucky enough that they're in, a monoculture, or they're in the wrong state, or they're attracted to other plants, and they miss out on that. So we're really keen to tap into that. It's been just super fascinating. There is a rabid, I'd say, group of researchers really good at this. Some doing it much longer than my lab, and the idea is to look at those plants. Plants aren't cognizant beings. They're not saying, "Hey, let's pump out some medicine for the bees that just came by." So it's not like it's an easy search where every plant is doing this or things like that. It's expensive maybe for them to make these compounds. And they probably make them for other reasons, maybe to defend themselves even against caterpillars. So our goal has been to look at that literature, look at a lot of bee ecology literature, but then really simplify it. So I think this, to me, a lot of research, you can do pure, like pure math, as it were, or you can be an engineer. And I've trended now towards the engineering side of it, where I look past some of the mechanisms, some of the, maybe even predictive qualities of things, and just figure, well, if we get a good test for this and we can deliver it to bees, we'll find out really quickly if those bees, if it's toxic to the bees, of course, and or if it's inhibitory for one of their diseases. So we really jump to more things done, I hope well, but at a very small scale. We use a lot of like plastic dishes with bees, petri dishes, and 8 bees at a time. And it's just, we have some running now. We did 200 bees earlier today, and we've done some other experiments with 400 bees a day, divided up into bunches of little replicates. And it's really a great way, I think, and maybe not just for this question, but for other ways to look at bee health in respect to pesticides and that. And other researchers are certainly doing this. So take those tests, screen dozens of things, and we're really down to sort of 10 molecules / extracts that we just keep coming back to because they're consistently doing something. And so I think we're at the point now where we're kind of, do we blend those together? Do we really pick one and make a claim on that one, or give it, tell beekeepers to plant that plant or something like that? And so we're really close, I think. And it's been, to me, it's something I devote half at least of my thought to these days.
That could be a citizen science project.
It could easily be, and I think there are people thinking that way, and I think think of all the master gardeners, all the city gardeners.
Yeah, that would be interesting. Way
commercial, farmers for that matter, forage, and then the beekeepers who are savvy enough to kind of track maybe a certain bloom that has these qualities would be really cool. So yeah, there's a potential for it to be a sort of rise up thing where people go and figuring out how to get that into their bees on their own. That would to me be really cool. It'd be cheaper than all the tests and labeling and producing. But the labeling and producing, if it's needs to be, and bee products, of course, as you know, that go into hives should be regulated. So if it needs to go through all that, that's another route, certainly, of course.
But if we're talking about a specific plant that you want to have an effect, very likely is in a field condition, you're going to need a number of people working with that. There's going to be astronomical. And now something that crossed my mind right now. What would you do, Jay, if you have 26,000 people like my channel? Each one of these beekeepers would jump in to do a little experiment in their house, that would could coordinate. Do you think? How can I help with the research with my social media reach?
Wow, that's a great offering.
You know,
Why don't we mull this over till before spring? And then again, there are other people. Melanie Kirby is a great scientist,
yeah,
bee breeder in New Mexico, is really into the plant side of this, like particular plants that might be attractive to bees. And so I think we're close. I really do. I don't know that I would offer up a natural experiment, for your followers right now, because
yeah. But if you have the idea,
yeah.
let me know, because
Okay.
I'm really into this right now. It just crossed my mind. Wow, I have this possibility here. If we coordinate very well with, I don't know, don't need to be the 20,000, but if you get 1,000 people there, distribute in different environments in US, in United States, and we just, simple experiment, but we increase the number of people,
Yeah.
that could be interesting.
Oh, for sure. No, incredible. And that honestly, that might be the quickest way to get something like this out to help bees and beekeepers. So yeah, I mean, not, just in general to have a grow your own sort of strategy on it might be.
Why I didn't think about that before? Why I didn't think about this before? It's something that I can, work together with the people that following, and we can do research together. Yeah. Uh,
I can connect you with a nice community of folks who are all kind of thinking that this is going to work out. So we'll try to make some headway.
Jay, since you are here, I need to ask you something. What's going on with the genome field of honeybees? What's going on in that area? What?
Oh boy, yeah, so the honeybee genome, as you know, was kind of first mapped out in 2006. So it's going on 20 years.
Yes.
Lots of promise. I think we did learn a lot about behavior and maybe immunity. And then now it's finally, I think, being applied. And this is in the U.S. It's through colleagues in Baton Rouge, Louisiana, for the USDA, and Brock Harper, who's at Purdue University, and a few others who are saying, we've had this tool for a while. We want to use it to really map out defensive traits, survival traits, and breeding integrity, I guess I would say, in some of the honeybee lines. And they're doing it. They really are. They're using the genome to validate Hilo bees and Russian bees, all these sort of bees that are high quality, lots of work behind them. They need it.
Who is doing this again? Sorry.
In USDA, it's Dr. Ariana Abalos, who's at
Okay.
and Lainey Bourgeois, who's also there as a research leader. She's done a lot of great work kind of mapping out the genetics of Russian bees over the years, but they're now using the genome to do so. And that whole lab, as the lab focused on breeding, it's getting behind this, I think, and looking at, how do different lines react to disease, and what makes the focus there, of course, has been on mite resistance. So what makes a lineage of bees knock down mite numbers, more or less, and I think it's gotten to the protein level for sure, where you can, whether you can harness that by traditional breeding, which is the goal, is just to use traditional breeding, but to kind of mark those traits as they go into, artificial insemination, things like that. And I think it's really there. I think they are improving certainly the integrity and the sort of the following of Russian bees and the now Hilo bee, and that's important, because you want to make sure you're getting the bee that you think you're getting. So you're doing that. So that's one big branch. I will say that honeybee genome was also the trigger for for Dr. Moran and others to see a bunch of microbes that we had no idea were there. So we work on a trypanosome parasite now, and that just kind of came out of genomic sequencing, and Like,
Go
well,
ahead.
that looks kind of interesting. And it turns out to be like nosema. It's sort of a energetic drain. It's linked to colony losses across the world. So it's something that we just didn't appreciate until the DNA from it hitchhiked its way into a study focused on the bee genome, so, and the transcriptome and stuff. So I think that's been a big plus, is just understanding the hitchhikers in bees, the bad microbes, the good microbes. There's great work on hygienic behavior. Of course, started with Rothenbuehler and then Marla Spivak, and now that continually gets more refined, I think, both with the behavior assays, but also the genes involved, and kind of really knowing what that series of events is to perceive. Whether it's Varroa sensitive hygiene, where they're actually sniffing out mites directly, or the impacts of mites on larvae directly in pupae, or just the general heightened awareness, as they say, often tied to Scutellatta bees, where they're just, nothing's going to get past them, whether it's a speck of dust or a mite. So those all have genetic components. When they differ between, one lineage of bees and one patriline, if there's bunches of drones in that colony, but when there's a striking difference, the same way with bee body colors, that reflects a gene or multiple genes that can be tracked, which is really exciting. Another pitch, so the honeybee genome is churning along, making a lot of data and insights, hopefully. There is a US effort, both with USDA and a bunch of universities, University of Illinois for one, to sequence the genomes of more of the U.S. bee population, especially focused on ones that are important for agriculture, from bumblebees to Osmia and Thaphora and some of the neat bees there. And we have a kind of a computational role in that. Most of the bee diversity is in the West, and most of the bee diversity scientists are at Logan, Utah, and Utah State University and USDA. And so there's this kind of big consortium effort now to understand different genomes of pollinators, and that, in our case, as honeybee fans, that should feed back to understanding the stresses faced by honeybees, having a reference group from other bee species. But those bees are important, of course, in their own right. So there's a lot of kind of neat discovery going on this year, especially on those genomes.
Cool. So a lot going on in that field too. And maybe last, just because you're here too, what's going on in the Beltsville lab? What those folks are doing? Something exciting going on over there?
Yeah, there's a lot of neat work, but some of it we work together. I work with Dr. Chen on the virus side, and she's got her own kind of neat work going on with knockdowns of viruses, and some diagnostic tools for viruses. She and relatively newer scientist, Dr. Mohammed Al-Baraki, are also re-upping our disease diagnostic service in ways that that never got done. We always thought about it. We're very proud of that service, but they're doing a lot of cross-year comparisons. Currently, a kind of a stream of state-by-state comparisons across the decades, which is kind of fun. Maryland got their moment recently, and in bee culture, and so there's kind of looking for insights in the same way the late great Bee Informed Partnership was doing, and hopefully some incarnation will do again, is to look without bias as much as possible, to look across time and states and try to see what challenges our people are facing. So that's been really fun. Dr. Miguel Corona in the lab is, of course, focused on nutrition, but also working with some pesticide stress now, and colony health studies. He's a very good beekeeper, and so he and Mohammed are doing some really neat kind of longitudinal studies. They had a paper on different hive bodies for beehives and temperature regulation, but they're doing some neat stuff, I guess, with monitoring hives, but also seeing how nutrition and other queen types and other traits interact. And then the holy grail for much of research is Varroa, and trying to knock back that scourge of honeybees, and Stephen Cook in the lab is kind of our lead on that. So he's tested dozens of miticides and worked with researchers who are, either testing the old miticides in new ways or new miticides in new ways, and also with the eye to push those through. And it's very clear regulatory pathway. If you're targeting a pest like that, you need to get a label. You need to go through, in most cases, EPA and try to work that out. So they're aware of that and doing all the right experiments to move the ones that are most promising out. So they'll get a stamp and they'll end up being part of the arsenal that beekeepers can use.
Cool.
But that's, yeah, we're all, we overlap. We work together in many ways, but our hook, we're not really bee breeders or even I talked about plant compounds, but I'm certainly not a horticulturalist, and I don't even play one on TV, inside the hive TV. So we have our focus is really on medicines, I would say, and management tools for beekeepers, and that's yeah. So we do overlap with each other on a lot of those projects.
But I'm sure people know that the bee lab and all the USDA's bee labs are doing a fantastic job. Actually, the USDA is doing a much better job now. I was critical of the USDA allowing outside people to see those things, but now there is a YouTube channel of the USDA. They're doing a much better job, and people can know the work that you guys do. In the field, many times I got the situation that people come to me, USDA is supposed to do this, and I said, well, they already did, many times, but people didn't know. And is this communication of the USDA with the outside world, but now they're much better. So I want to congratulate the USDA people about their communications. Jay, I just want to say thank you one more time for your time, for the work you do in the name of our community here. We always appreciate when people come here and share their thoughts, the research areas, and we only have one goal here is to improve the beekeeping industry. And in the name of the InsideTheHive.tv community, I want to say thank you.
Yeah, thanks so much, Umberto. And you've always been a friend to bees in the bee community and to all of us personally, as from your time in Beltsville. So it's an honor to be on your show, and I love what you're doing here. Your positive messages to improve bees and beekeeping, which is a great thing.
Okay, that being said, thank you, Jay. And I'll see you guys in the next week. InsideTheHive.tv, the show about bees. See you guys later. Bye-bye.